Okay, fine. So let's begin the final session of the day. And once again, we have Chandrasekhar with us, who will be giving part two of his talk. So Chandrasekhar, over to you.
Thank you, Shruti.
Oh,
So,
oh, yeah, and if time permits, he will be giving like a bit of a tutorial as well. Yeah.
if people want to just take a look at the codes that I use in the simulations and so on, I'm happy to share them. But you know, it's not often you take a look at a code unless you need it. So
Okay. So go ahead.
yeah. All right, I'll share my screen. All right. So I guess welcome back. So today the idea is to sort of talk about how you go about modeling cell migration into in couple of specific biological context. Um, Before that, I need to get rid of this. One of them is something that I worked some time ago. I've been so it's more complete. The other one is something that I'm actually thinking about. But I think the intention, at least my intention here, is to sort of give an idea as to how I think about sort of reducing the complex biological context to something that is uh, simple enough, like the toy model versions of those same thing, and get some insights into the phenomena. That's so. I hope as long as give some insight as to how I think about it and how one thinks about reducing the problem to something that is you can, you know, computationally track, and that is what I want to sort of convey. Okay, the first one is something that I've been working for a while. It's called the germ cell. It's with germ cell transepithelial migration. What exactly is the biological context here? I don't know how many of you know this, but in Drosophila, in Drosophila melanogaster, to be fair, it's in all sister species of Drosophila as well. The pole cells are formed very early. The pole cells are what are basically the germ cells. It's the primordial germ cells. So they eventually become the germ cells. So, but they are formed very, very early. And they're called the pole because they're actually pole cells because they're formed at the posterior tip of the embryo. But right after gastrulation, for people who don't know what gastrulation, if you have seen these famous videos of Drosophila embryo development, you would have seen at some stage there's like this huge churning of the embryo and a lot of things just start moving and there's a huge amount of flow and so on. So, so that's the gastrulation. And right after gastrulation, you just see that those pole cells have been sort of displaced into this sac, an epithelial sac called the primordial midgut. And they're stuck there, which is shown in A here. And by the end of embryogenesis, by stage 16, that is, they are supposed to go in two different batches, actually, because there are two gonads. They're supposed to go to these two gonads and reach there. So there's quite a lot of long journey that to take. They start from the posterior tip. After gastrulation, they are somewhere inside the midgut, which is an epithelial sac. They're stuck inside the midgut, and they have to exit this midgut, which, to remind you again, is an epithelial sac. There are no holes for the germ cells to exit. It's a sac made of one layer of epithelia. So they have to exit that. That's one of the crucial parts. And in Drosophila melanogaster, we know that out of around 40 pole cells that are formed in the beginning, only about 66% of them make it to the gonad finally, which means there's quite a lot of cell loss. It's an active area of study as to what determines your success rate, blah, blah, blah. But not being able to exit the midgut is also one of the major places where some of the germ cells just don't make it and accounts for the 34% of the germ cells that are missing. So this is an interesting instance because when they arrive in the midgut, they're a clump of cells which are sort of connected to each other, but they exit the midgut as individual cells. So as far as exiting is concerned, it's a single cell migration. It's as they never are connected to each other while they exit. So it's like a single cell migration as far as the exit is concerned, which is nice. I'll tell you why it's nice. Okay. I mean, I don't know how well, I mean, I learned one thing, which is that there's microscopy and then it's actually takes a while trained eye to sort of even appreciate some of the microscopes sometimes, unless it's too obvious. So what I want to show in this below figure here is that I don't know whether how well you can see this sort of um, lumen of the midgut, which is the gap inside the sac and the bunch of germ cells that are exiting. Right next to the label A and so on, these are the germ cells that have already come out, and then there are a bunch of cells that are stuck inside. So, okay, something easier for you to probably understand is something that I do. So if you see, this is the mid, I know this is like, if you look at it, um, this is an instance of some of them having already left. It's a schematic showing some of the germ cells leaving, uh, leaving at different stages of basically the germ cell exit. And as far as the germ cell and the epithelial cell are concerned, how do these guys basically exit when there is no gap? Because you're going through an confluent tissue. Uh, there are like initially some hypotheses saying that there are always these junctions between the epithelial cells are very dynamic. There are like these transient gaps that open up and the germ cells can squeeze in. It's a bit too hard to believe because germ cells are not tiny, as you can see from here, and the gaps that form because of dynamic, the junctions being very dynamic is not sufficient enough for them to just squeeze through. Everybody agrees that there's a pull force that is pulling them outside, but the gaps are never that big. Sorry, that I think I interrupted a question. Did I not? Okay. So so when they so our hypothesis was that uh, this is an active, I know the germ cells actually form E-cadherin junctions with the epithelial cells because epithelial cells naturally have E-cadherin because the E in E-cadherin comes from epithelial cells. So the epithelial cells bind to each other through E-cadherin. And experiments also showed that germ cells also have E-cadherin on their surface. So, and our point is that, it's not these things, the E-cadherin on the germ cell is not just incidental, but actually is functional. It sort of makes these transient connections with the epithelial cells, and that is what allows them to exit. So, in order to test our hypothesis, so we wanted to obviously reduce everything to 2D. So, if you take the sac and slice it in certain angle, then you can basically get a circular ring of epithelial cells. And conveniently, because the germ cells exit as one by one, I mean, as individual cells, we can just understand this process by just taking one germ cell exiting. Maybe there are some aspects of it which requires you to basically study multiple cells exiting at the same time, but for much of the things that we are interested in, we just we can just consider single cell exiting an ensemble of this for different parameters and can get some insights into the whole process. And that's what we went on to do. So what we ideally want is a ring of epithelial cells and a germ cell somewhere in the middle, and it sort of and there's a radial pull, and these things exit. The problem is, in the green cells, which are the epithelial cells, they obviously have very strong adhesive affinity. If you try and just basically say that, make this ring, you can think from what I've told you and I mean, from yesterday, these things will collapse and it won't be a ring anymore, but a clump of epithelial cells because energetically it is more favorable for them to basically overlap on top of each other. All right. And to maintain this lumen, they're just increasing the number of pixels at the boundary that are not interacting with the epithelial cell, which is not energetically favorable. So the lumen will just collapse. which is a bad idea. I mean, which definitely doesn't give us what we want. So the one way to do this is the lumen that I show is not medium, but an actual cell which has the same properties, adhesive affinities, and everything. As far as all these adhesive affinities are concerned, they are like very much like a medium, but with one condition, which is that unlike medium, it has a volume constraint. So it's incompressible in some sense. So there's cyan colored stuff is this cell. So once you add that in between, now we know that that can prevent epithelial cell from collapsing. Nonetheless, that's not a guarantee that the epithelial cells will still remain in a ring, because they can just all detach and form a clump while leaving the uh, cyan cell somewhere else. So the way people basically fix these things, because there are certain geometries that are harder to achieve because it's actually going against energy minimization. But this is a precondition, and this is an artifact of, like, and to be fair, this is not completely an artifact, because often there is ECM, um, and this lumen is not nothing, it's not void. And two, it's very hard to account for like a certain pressures that exist within a 3D structure and so on. So the way we do this is by finding a hack which does this. One thing that is very convenient in cellular pods model, to be fair, this is not a feature. Well, CompuCell 3D readily provides you this, but again, this goes into something that we spoke yesterday. We can establish what is these things called, what CompuCell 3D likes to call it because of focal point plasticity. I think that came because someone who used it for the first time in a paper probably called it that because it was that is what they were trying to get at. But what it effectively does is that you can connect centroids of cells through springs and have a quadratic energy, and say that there is this resting distance that I want to maintain between the centroids of these cells, and I can obviously have a spring constant. And I can also have another parameter which says that, beyond a certain length, if it really expands beyond a certain length, then the spring just breaks. And if you do that, then you basically can get this nice structure. So here, if you see all the white lines, like every neighboring epithelial cell has this sort of spring connecting them. And not just that, but all of them also have a spring connecting to the center of in this cyan colored cell, which represents which prevents the epithelial from collapsing. So then you get this very stable structure. And in this 2D epithelial ring, then we perform simulations and try and understand what is the role of adhesive affinity between the germ cell and epithelial cell.
Hey,
That's what we set on. Sorry.
yeah, sorry for interrupting, but I have a question. So in the biological system, what's exactly preventing the epithelial cells from collapsing, like you mentioned before, um, which would happen in this model if there was no support structures?
So the thing is, one, very often inside of the lumen, the cells actually have um, an extracellular matrix and stuff, which is basically a mesh that sort of keeps
Right,
things
right.
from collapsing. So yeah, we know that is the case because we don't see the lumen collapsing, at least in normal cases. So we know so this is a thing that we want to achieve because that reflects biology. How we are achieving it is not probably how biology is achieving
Exactly.
it, but the idea is to make sure that whatever that we have built here doesn't affect with the process that we are studying.
Okay, because
Yeah, but that's what.
at the end you don't want the lumen to collapse, right? You want the lumen to persist. But
Yeah,
then
yeah.
where you're doing to the model so that the lumen is persisting should be actually physically, should have some physical basis in the biological system as well. So I just don't know how exactly do we draw lines between that and this.
See, okay, I get what you're saying. Like, if ideally we would want the lumen not collapsing as an emergent phenomenon of something that is, let's say, largely justifiable, or like, something that is more justifiable in some sense. But all I said is the reason you do this is because there are a lot of components that allows the prevents the lumen from collapsing in the actual system that we don't model. So we take it for granted that this thing is a stable structure, and we just do whatever that needs to. We add these constraints to make sure it doesn't collapse. It's like we take it for granted based on what the biological information. I agree. I mean, it's not organic, so to speak.
Yeah. Okay. Thank you.
Yeah. No, but I agree with you. Like, you can probably just go find a way which is more justifiable, like, find a way to sort of, get something like an ECM. But to be fair, I can, if you allow me to just argue, I know that if I remove the cyan cell, the system still doesn't collapse. I know this.
Okay.
So the spring alone is enough. So what exactly is the spring doing, right? It's like basically a mesh of things that actually connects cells next to each other in some sense, right?
Yeah.
So it's roughly doing that, but then again, it's very hand wavy. I mean, yeah. All right. So okay, this is probably a point I need to mention. So long back when I was actually simulating this thing without copy cell 3D, I used to do this bit differently. I used to prevent this ring from collapsing very differently. I used to draw a region in space where the epithelial cells interaction with epithelial cells interaction with the pixels belonging to medium was low only in this ring, and outside of the ring it was very, very high. So none of the pixels in the epithelial wanted to leave that ring, so they just remained as a ring. Okay, I don't know because because I could do that, whereas in commercial thread it's very hard to have a spatially varying value for a J value and so on. So I did this. Somehow I must say that I was very annoyed that I couldn't do what I did when I was writing my own code. But I would say this is probably slightly more justifiable given that the springs are probably a much closer approximation to like what ECM probably does rather than anything else. So
Right.
yeah.
So if you use different methods to maintain the lumen, so at the end, do the outcomes vary in some sense? Like, you might not have done it, but do you have any idea about that?
Yeah. I mean, so the thing is, outcomes vary uh, slightly quantitatively, but qualitatively things remain the same because
Okay.
I've done the simulations in both the cases and I know what it looks
Okay.
like. Yeah. So that's a good thing because end of the day, it's very hard to connect the exact values of this J to actual biological parameters. You can only hope for like qualitative, dependence on like we can only make qualitative statements, and in that sense, like something happening at slightly different actual value of some parameter is probably not something that one should worry so much. So the exact nature of how you implement this does have an effect, but not drastic effect, to answer your question. So this is okay. Let me just play instance of where the germ cell manages to exit. Okay. And this is an instance where, well, it doesn't exist because I know, or at least for however long I have simulated, it doesn't exist. And what is long enough? The answer is in development, like it's a series of events, and like you don't have forever. Like if something happens, like in an extremely long time, it's probably as good as not happening because things would have moved on to the next stage of development. So in some sense, where exactly draw the line? These are the things that one has to really worry about because how do you connect your Monte Carlo steps, which is how you measure time in your simulations, to the actual developmental time, are some of the things that probably end of the day, one has to think about a lot. But one way I do it is I run for sufficiently long and then say that, whatever I see is not because I didn't run it for long enough. But as you can say, but that's not the best justification for things, because end of the day, it's too hard to go from the Monte Carlo steps to biological time. So the only way one can possibly do is to sort of, find some uh, process whose time scale is shorter than the phenomena that you're studying, and sort of try and try and represent the whole dynamics in terms of that, so that uh, uh, you can connect these two different time scales.
Hi, I had a comment. I was just wondering. Like for some of these cell types, I mean, we would know we may have some idea about the rate of I mean it's diffusion coefficient for example, right? We would know things like that. And we would know, I mean, we would know the size, and we would have some of these physical measures, right? So based on that, could we use that to sort of match up the time scales in terms of like comparing the Monte Carlo time steps with the actual sort of time that's elapsed? Could something like that be used, for example? I just wonder.
In some convenient cases, yeah, but probably here there's nothing I can actually, because I know the typical in a wild type case embryo, how long a typical germ cell takes to exit. I have that. Um, I have that, but uh, still, nonetheless, when that itself is the quantity that you're trying to measure as a function of other parameters of the system, it gets very tricky. It does. I mean, honestly, this is where um, you can observe different regimes and different parameter, different phenomena, different things that you see in in the experimental system. But ideally, would like to basically have a correspondence between the parameters that you use and the biological system. But that's hard, especially in the cellular pulse model. That's pretty tricky. Very often, very tricky.
Okay,
That's
thanks.
unfortunate, but yeah.
Thanks.
Okay, just to make what is more important is that very often you can actually get relative strengths of the parameters. It could be the relative affinity between adhesive affinity between, let's say, two different cell types or something. I think as long as we talk about ratios and relative strengths of two different things, I think we are in better shot at like trying to test establish a correspondence between to the biological system. But absolute numbers almost has no meaning. All right. So what we did was we wanted to so the two things that we wanted to sort of vary and study the system was that one, the strength of the chemoattractant that is pulling the germ cells outside, and two, the adhesive affinity between the epithelial cell and the germ cell. So we do that, and we found something very interesting, which is that, of course, if the strength of the chemoattractant is very weak, then you just don't, the germ cells don't exit. On the other hand, when it is very, very strong, it exits with very high probability throughout. I mean, almost almost independent of the adhesive affinity between the germ cell and the epithelial cell. But for a lot of intermediate values, you will see that it's very non-monotonic and has a very clear peak. There is an optimal value of the adhesive affinity between the epithelial and the germ cell at which you see the maximum P exit, or that also corresponds to regime where you have the shortest time for exit. So not just we see that, but it's what was pretty interesting is that this particular value is almost independent of the strength of the chemoattractant itself. it was independent of the strength of the chemoattractant, but rather just, that optimal value. That's what I've kind of shown in this red shaded region. The peak sort of nicely aligns. So we were like curious as to, so what exactly, I mean, so is the adhesive affinity between the germ cell and epithelial cell, like the optimal value at which, optimal value is independent of the strength. So what is it dependent on? I mean, does it depend on so we had a thought, we suspected that probably what determines is the relative strength of what epithelial cell affinity is and the epithelial to germ cell affinity is. So because in this simulation, we kept the epithelial affinity the same for when we varied the other two parameters. Maybe this is why the optimal value sort of doesn't depend on other things. I mean, okay. And just the figure on the right side is just basically showing you the trajectory of these germ cells in 1,000 iterations. One, the figure at the top is when most of them have exited, and figure at the bottom, but quite a lot of them didn't make it. And as you can see, the trajectory is actually concentrated around the gaps between the epithelial cells, because that's exactly where germ cells can basically get through. So, so we went on to see, okay, does the optimal value of JEG depend on the JEE if one were to vary that, keeping the strength of the chemoattractant at a fixed value? Do we see dependence on JEE? Just to test our hypothesis that, what actually matters is the relative strength of the affinity between epithelial and the epithelial germ cell. So sorry for calling P exit as P success in this figure, but it's the same thing. But if you do that, you'll see that it does depend. On log plot, you can see that it does depend, but it's not it doesn't increase very strongly with it for a good range of values. It's actually slowly, did someone have a question? So you do see this sort of weak dependence on this, but nonetheless, it does depend on JE. So in some sense, one can say for a good regime, good range of values, the optimal value at which the germ cells exit seems to be almost even independent of JE to an extent, but only once the JE goes up, significantly, then you do see some change for the optimal level of JEG as well. All right. So, of course, the part that I'm not going to talk about because is basically how we test this. So I have an experimental collaborator who basically one thing that we can actually tweak is the level of germ cell expression. Epithelial E-catenin expression on the germ cells, and see if we increase it a bit too much, or what happens to the success rate of them exiting. So that is something that we can reasonably do. Experimental detail that I don't want, just want to sort of tell you is that in Drosophila, it's slightly annoying because these are all early stages of development. And the problem with Drosophila is that a lot of the things in the early stage of development is all dictated by what the mother has deposited. A lot of the mRNAs are rarely transcribed by the cells, but rather they are just inherited from their mother and they're just making produce out of that, which means it's very hard to change the expression at very early stages because they are not actually engaging in transcription to make the mRNA, but rather something that they're just working with what the mother left. So you have to alter much early. But then if you alter it much early, then you might be affecting processes that are prior to what you were interested in. So this is what makes it a bit tricky to alter these things when you want to study these things. So any sort of test you do is going to be not very clean because of this because of biology. All right. So we saw this instance of from the Drosophila development, which is even though there are a bunch of germ cells, they exit as individual cells. So let's take a look at another system that I'm kind of interested in sort of actually working on. you know, from very recently, which is this border cell migration. It's a fantastic system. And this is also a very cool thing. If you look at this image is actually of what is an ovariole. Orioles are the substructures that form the ovary. So an ovary is consisting of multiple ovarioles. And they have this very beautiful, so each of these entities that you can see is an egg chamber. And it's not like I've arranged things from different, I mean, they have arranged things from different times. But rather, this is exactly how it's within. This is almost looks like an assembly line of the egg chamber. If you open up an oriole, you will see this nice ontogenic series of things that are like in much early stages and much later stages there. And that's because like something on the on your right side, as you can see, as the embryo that is in a oocyte that is close to completion, and that will exit, and after that all the nerve cells die and the egg chamber goes away, and the next one basically produce, the next embryo is going to come from the one that is before that. So it has this nice beautiful ontogenic series within an ovariole. The process that I'm interested is something that is highlighted in the early 9, and as you can see there's like 2 arrows there, which is a cluster of cells called the border cells. So border cells are essentially, so the egg chamber is lined by these cells called the follicle cells. Among those, I mean, about 6 to 10 of those follicle cells sort of achieve this different fate called the border cells. And they enclose these 2 special kind of follicle cells, which are called the polar cells. And this cluster of border cells enclosing these polar cells actually do this beautiful migration from the as you can see, from the anterior to the posterior of the egg chamber, and basically stop at the place where the oocyte is. And this migration is like around, they travel a distance of 150 micrometer in a time of like 3 to 6 hours. And it's a great system to study for multiple reasons. One, it's a great system to study collective migration. What is fantastic is that it's an instance of collective migration, but unlike wound healing and so on, this doesn't have like hundreds of thousands of cells, but rather just there is like 6 to 10 cells with 2 polar cells. And this is not like an inconsequential cell migration, because the polar cells reaching there is essential for the oocyte to have polarity. So it's very important. Whatever the cargo, the 2 cells that carry is super important that it reaches there. And they have to travel through these nerve cells, which are these bigger cells, which are 15 cells, about 15, there's 15 nerve cells. And they go between these nerve cells and reach the oocyte. And the oocyte secretes something that is basically attracting this cluster of border cells. So that is a contextual problem. But the beautiful thing is this system can be ex vivo imaged, which means that you have very good images. So this is from the previous group I mentioned who have imaged this. And they have labeled, they have basically put green fluorescent protein in the border cells. So you don't get to see polar cells, but you only get to see the border cells moving. And you can see that they actually start from the anterior to the posterior tip, and you can see that they go between the nerve cells and reach there. At least that is the case when things have worked out. Okay. E-cadherin is super important here because all the 3 relevant cell types here have express E-cadherin, but in different levels. So the border cells express a reasonable, the polar cells have the highest, the 2 red cells I highlight inside have the highest amount of E-cadherin. And the border cells that enclose them have the second highest. And the nerve cells, which are the cells through which they go, have the least, but nonetheless, they do have E-cadherin. also highlighted in the cell in the very front called the leader cell, because that's a point I'm not going to make here, because they also do something very interesting, which is the identity of the leader cell keeps changing. They keep taking turns. So it's like not just they're moving, but they're also, which border cell is the one leading keeps changing, which is quite interesting. But the key point is they go from one end to the other, and they perform this chemotaxis in a very, very crowded environment. There are no gaps here. They're just going through nerve cells. Okay. But they've also done something very interesting here. You can change the E-cadherin levels in these three cell types. And I don't think anyone bothered to do it in polar cells, because polar cells are like well within, they're always enclosed, and then like they're kind of almost passive, like carried by the border cells. So when they decrease the nerve cell E-cadherin, I remember nerve cells have the least amount of expression already. When they decrease it, the germ cell, the border cells cluster kind of fail to migrate, or at least they get lost, and they most of the time they just don't make it. When you increase the nerve cell E-cadherin, again, we have increased the E-cadherin levels in the cell that express the least, and they always make it. It's just that they're slower. They take a while to reach there. Similarly, when you do it in the border cell, when you decrease the level in the border cell, which has the intermediate concentration, it does affect the migration. But if you increase in the border cell, it makes no difference. If you think about it, it should make complete sense, because something that I pointed out yesterday is that when you're making these junctions, if you argue that the number of junctions you make is they're crawling on top of each other. The germ cells, so these border cells are actually going crawling through nerve cells. And the crawling process involves making these junctions, adherent junctions, using the E-cadherin. So the number of E-cadherin junctions that you can make, assuming this process of junction formation is very quick, is limited by the cell that expresses the least number of E-cadherin. So if you increase border cell already has more E-cadherin than nerve cell. If you increase the border cell E-cadherin, it should have no consequence, because you are not limited by the border cell. The rate limiting step is the amount of E-cadherin molecules on the nerve cells, and it kind of makes sense. On the other hand, when you express it a lot on the nerve cells, it should not affect, but it takes a while. You're just making more connections. And I will argue why making a lot of connections is also not very good, because understand, like crawling involves making these connections and also severing these connections when you have to take a first step further. Not being able to make any connections is bad news, but also making a lot of it also slows you down. So as far as you can imagine, as far as the cellular pass model is concerned, the adhesive affinity is all represented using this J matrix, where between the different cell types, you can assign a value that determines the adhesive affinity. Okay. So I'll show you just one simple instance of because the nuances in the shapes of the nerve cells and stuff is not very relevant here. Let's just consider one gap. It has this two blocks of nerve cells, top and bottom, and these guys are going through. This is so the blue cells here, there's one massive blue cell at the top and one massive blue cell at the below. I'm just representing them as the nerve cells above and nerve cells below. It's the simplest 2D model you can consider. And the germs and the border cells are the green cells, and the polar cells are the ones in red. So I just want to highlight that when I basically reduce what is equivalent to reducing the E-cadherin levels on nerve cells is to basically increase the J values for the border cells with the nerve cells. When I do that, you can see at the below, everything is identical except for where the nerve cells have less E-cadherin in subsets. The whole thing goes much slower, which makes sense. All right. But it's also important that, from my previous work on the germ cells, it should be obvious that, the strength of the chemoattractant is also relevant. So which I've not explored it here, but I've kept it as a constant. I've basically only changed this, and you can see that why things slow down when you basically have no E-cadherin on the nerve cells. It just the migration slows down. So both these things basically gives an idea of like, when these migrations that necessarily involve making these connections and moving with adherent junctions, and like you form adherent junctions, it's like how you climb a ladder. You climb a ladder by basically attaching your hands or palms or fingers to each of these steps and use the traction to go one step above, and you also need to basically leave your hands to go further away. Let's so one way to look at it is that if you take a kitten walking on three different surfaces, a first surface is where there is no E-cadherin on the surface that you're walking, or in other words, a very slippery surface. There is no traction. You can't move. That affects your movement. B is the intermediate level of traction where it's like a carpeted surface you can walk. So the nails stick in, you get traction, you can take it out and keep taking a step further. And the case where you have too much E-cadherin is like bit like, I don't know, some gooey American melted cheese, like processed cheese on the surface of the carpet, which is like a lot of traction, but it is every time you put your paw there, it takes a while for it to detach from it, slowing you down. So this is what I think as far as the crawling with making these adherent junctions imply, which is that there's an optimal level of adhesion that is needed for movement. Too little is bad, too much is bad. Too much usually means it slows you down, and in slows you down can be pretty dangerous, pretty bad in development, because everything has to happen within a finite window, because there are things that are following, and if you don't make it in that time, it's probably as good as you're not making it. Okay. Does anyone have any questions? All right. I just have one small piece to cover, and then probably if you have questions, you can ask after that. So this is something that I tried. Sorry.
I have a question,
Go ahead.
So this non-monotonic dependence of cell motility on adhesion is interesting. So do you have a curve of this? I mean,
The one place where I've explored enough to have a curve is this.
Okay.
But border cell is very interesting, because there the experiments have indicated that in some sense. I mean, it's not the problem with the biology is that, I mean, not problem with biology. It's limitation of experiments that you can't do this fine regulation of E-cadherin. You can do this knockout or an extra expression. So it's only very few values that you can explore in some sense. I mean, if there is a nice way to there's a fine way to tune the knob, like you have in simulations, where you can change the expression level of E-cadherin in these 3D cell types nicely, you could get a nice curve experimentally as well. But sadly, experimentally, you can only do like, oh, I made this expression, I'm over-expressing it by, making more copies of it, or I just knock it out, which is like sort of, you can only, sort of explore few regimes. So it's a bit of a limitation, but the border cell, I think there's quite a lot of experimental support that there's an optimal range.
And also, is it something well known in the community, this fact, the dependence of cell motility on adhesion? Or is it just something that you're finding it for the first time in your simulation?
No. Absolutely not. People do know that the E-cadherin is necessary for like movement. So it's like, one wouldn't go on to generalize it. Like it's like, I guess the problem is that sometimes people are interested in their pet problem. They like take a nice phenomenon and they try and study everything, and they study E-cadherin as study the effect of E-cadherin, blah blah blah. But very rarely they want to generalize outside of the context, because but E-cadherin being relevant to cell migration in border cells at least is kind of well known.
Non-monotonic
Why
this non-monotonic behavior of motility with respect to adhesion, is there experimental evidence for this?
I would say that if you combine some of the results and across different phenomena, one would come to that. One would sort of make a case for that. But I'm not sure whether people have sat and made a case for it. But rather they probably made their observations in the context of the problem that they was looking at. But I would be shocked if people have done it. But like I'm just using Cellular's model to sort of make that as my key hypothesis and sort of verify it. But I do think that if you take the old literature and experimental results, that might be your conclusion as well without even doing anything. But then again, as I said, like, and I am very biased because I'm sort of I believe in this, so maybe there's another alternate explanation.
Okay, thanks.
I mean, it's very hard. I mean, it's very hard. I would like to believe that it's very novel, blah, 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 but then again, like, what is sometimes novelty is just me not doing enough literature surveys, so I don't know. Yeah, it's sometimes it's like people have reported in works where the focus itself was something else. So, but in somewhere, what is very important for you has been mentioned, but they don't care about it because they were worried, they were more interested in something else. So it's just very hard to do thorough literature survey. All right. So this is going to your question yesterday that, Philopodia. I was thinking about it and I was like, yesterday I made some simple simulation about, this thing. And I also wanted to highlight something because I thought this is a good opportunity to highlight something that is, super relevant, super useful, which is sometimes you want to break your cell into multiple compartments because it's very hard to consider the whole cell as an homogeneous entity. At the bare minimum, sometimes the nucleus is less, compressible than the rest of the cell. So you would probably in some processes that is relevant, and you want to have a subcellular region which is nuclear with a very different physical property. So sometimes you want to have a moving front that is much more wiggly and so on. So that's how, and that is something that Computational 3D allows you to do it. And within the CellularPods framework, you can easily do this. And just to show, like, just the simulation, like, this is something that I'm showing. Like, what I've done here is that the green is the moving front. That is much more wiggly because the J value for that region with the medium is lower than what the red region has. And below, it's the exact same thing, except that I'm doing it at a higher temperature. Okay. Is it a good proxy for Philopodia? I don't know. But I just wanted to tell you that, sometimes you can have a cell have sub-regions that have very different physical properties, and the way to do this is by compartments. But at this point in time, I need to
just one minute question.
Yes.
Yeah. So can you also take into account local curvature or curvature like
yeah. Like can you see my Computational 3D screen?
Yeah.
I think somewhere there is I don't know. I've never used curvature, but I think curvature is something that, some people in the past have used it. So I think they allowed for it. There's some curvature plugin or something.
So,
but you can do that.
So if my target surface area is very large and the volume is very low, and I impose the condition that, the surface is sort of stiff, so extreme curvature is not desirable. Do you think that could lead to these sort of branches and flagella-like structures?
Could be. Another thing which is a bit unfortunate here is that in my simulations, okay, let me go back to the simulations I was just showing. No, this is, I think, yeah, this is the one. So let's just see what this looks like. So I have this thing, the medium, and then there's a nucleus, and then there's a front. Sorry for the fonts. I mean, I just keep changing fonts every week because it sort of makes it easier to. I have weird font saturation, so currently going through a bad phase, I suppose. So it's just so this, but I find this font very appealing right now. But if you take this is the XML file that accompanies most of your simulations. And then there's also Python file where you can tinker and do things. And you define a cell by saying in this cell field, pixel from such and such is your bulk one, nucleus one, front one, because I'm making two cells. Similarly, you do this and you set your target volumes, you set your lambda volume, you can set your target surface, lambda surface, all the quantities that I mentioned. And I'm also saying, because this is a multi-compartment cell, you say this nucleus one, front one, bulk one, they together are part of a same cluster. So I give a cluster ID, which is the same for all of them. And similarly, I do a cluster ID for all this. The most important thing about this is that when you have multi-compartment cells, there is something very important that one is to take into, which is the energy values that you now have. There are two sets of them. One, when compartments, when these compartments belong to two different cells interact with each other, as opposed to when the compartments belonging to the same cell interacting with each other. Because so that's why it has this something called the contact internal. There's a contact energy internal. So you have to be very careful in picking these things. Sometimes you want your nucleus to have great affinity for the bulk of the cell, because you don't want your nucleus to leave the cell. But you also need to tell that, my nucleus has this affinity for my cell and not for other cell, because you don't want your nucleus to jump to another cell. So in order to avoid such goof-ups, and I'm not even taking a very complex system here, but you will realize that, then you need to, the moment you start introducing compartments into a cell, and you need to have this, energy parameters defined when things belong to the same cell, when things belong to two different cells. So that's something that comes up when you have multi-compartment. And much of one of the most annoying things about CompuCell 3D is to sit and basically tweak these parameters and see what it does. And this is where CompuCell 3D has a massive advantage, because I can just tweak this parameter and just like see what the output looks like. And this is where I think writing, writing your code as a code opposed to CompuCell 3D has this massive advantage. Basically, how quickly you can verify some of the changes and the tweaks that you make. The time scale it needed to sort of test these things is so short that you make much greater progress. At least that's what I feel. Because it has this nice inbuilt system to sort of visualize things then and there. Otherwise, when you write your own code, every time the visualization is also something that you do by hand and can be a pain. So
This was happening in real. What you just showed.
Yeah, this is happening in real time. It's pretty fast. Yeah. It's surprisingly very fast. So I just want to sort of go through this file just to let you know that everything that I mentioned, especially yesterday, and where does these parameters factor in here. Number of processors, debug output frequency, ignore that. That's very not important. Dimensions. This is the space dimension. Okay. So this does 3D. I'm not showing you any 3D, but if you change the Z value, then you have 3D space. Steps, Monte Carlo steps. Temperature. Neighbor order is basically like, you're defining the neighborhood. So how many neighbors next to each other. So, okay. Cell type. So there are 4 cell types. We're just giving IDs here so that you can refer to them. Okay. And this is the volume plugin, which I've used, which I'll show you what you do with it. This is where you impose the constraints and like the target volume and the strength of the constraint. Same way for the surface. This is something for the center of mass, because very often you want to keep track of the center, where is the center of mass of each cell with time and so on. So this allows you to sort of do that. A neighbor tracker is another plugin that allows you to know at any point in time what are the neighbors of each cell. Okay, this is the most important thing, which is the contact, which is the internal, and which is the energy parameter. So because it's an symmetric matrix, so you only define these number of terms, which corresponds to, let's say, the upper or lower triangle of your J matrix. This contact internal because now we are dealing with compartmental cells, so you have to like have two sets of these parameters. Chemotaxis, as you can see, I'm just saying there is an field chemoattractant named ATTR, and the front and the bulk of the cells are attracted to them, and the strength at which they are attracted are 100 and 100. This is the lambda. This looks more annoying, but it is not. It just basically shows you the field. So, in other words, if I switch to the ATTR field, it has this very clean gradient. I'm just now saying I'm making it extremely simple. I don't have any diffusion or anything. I'm just starting with an initial condition where the field strength is just proportional to the x coordinate itself, so that you have a nice linear gradient. So, but that's how you get the gradient. But sometimes if you don't care, there's also a way where you can have a file where you put the concentration in every point in space and just say, read the concentration value of this molecule from that file. That is also a possibility. And yeah. And focal point plasticity is this plugin that allows you to add these springs in case you need them. I haven't used them, but in case. I usually load all of these things because, sometimes you do need them. And here is where I mentioned that, where you mentioned the lambda V, which is the volume constraint, and say I assign the volumes for each of these three regions and say this is the lambda V. I assign the surface. I've basically taken what is the corresponding perimeter if it were a circle and given that as a surface value and put the constraints. Yeah, that's pretty much it. Of course, I didn't teach you like how to use Composite 3, but just telling you that, how things look, it's just sometimes good to see. And I think nothing that I've shown you should probably look very intimidating here. It's very clear in the Python steppable file. It's very clear for everything. So for this particular program, at the start, this is a thing that does things in the very beginning of the simulation. And at every Monte Carlo step, you can do something, which I'm not doing anything specific at each time. And when the simulation is over, it can do something. Or you can also have when you abruptly stop, it can also do something. So usually you basically add lines of codes that saves you some data that you need and so on. So that is pretty much what file that looks like. You can make it as complex as you want based on the context and so on. There's no zoom. Yeah. So in case you have any questions, please do ask.
So this Python and C++ files, I see. So basically in the software, you'll create these simulations, and in the back end, it will have its own analogous Python and C++ files, or you actually wrote the whole code for the Python and C++ version?
No, you don't. Unless you're trying to do something that this Composite 3D doesn't provide already, you won't write a single line of C++.
Right. Or Python per se. So basically, you will create a simulation using the software, and in the back end, you will have the Python code generated for that. And then if you want to tweak
No.
it, add any additional thing, no.
When you create a project, it gives you an XML file and it also gives a Python file. Okay, you tweak the XML file, you tweak the Python file if you need them.
Okay.
Hi, I had a question regarding the border cell simulation that you just showed. So you showed two cells that are sort of unattached to each other, that is migrating, let's say between.
Unattached.
So the border cells
Sorry.
were migrating independent of each other.
Where? Sorry. To go back here. This is the one you're referring to, right?
Not this one. You showed two cells
Oh.
that.
Oh yeah, so that was not a border cell. That was just basically just to show that you can make a cell into multiple components. Yeah, that is true. That is true. I just added two just to show that I don't know. They're actually migrating independently. Yeah, that's true. Yeah.
So in this simulation, what if the cells are sort of migrating like a cluster? So have you run the simulation? Sorry, go ahead.
You can usually do that by just tweaking the J matrix here. If you start pretty close enough and you basically create an affinity between the red regions or the green regions of the two cells, they will they will attach to each other and migrate together. Sometimes if it's a bit of an energy calculation, sometimes if it is actually more energetically feasible for them to just move on their own rather than form this clump, then they will probably form this group. They won't. Otherwise, they would just like, if there's sufficient affinity for these two different cells to bind to each other, then they will move as a cluster.
Okay, the reason I'm asking that is because typically when cells migrate as a clump, the clump is not very big. It's made up of a few cells. So in this case, it's a border cell made of some 7, 8 cells. Even if it is let's say a cancer cell metastasizing and moving on to a different site, it's usually a small clump that breaks off. So I was wondering, in your model, is it possible to test if you keep adding more and more and more cells, would the cluster sort of be inhibited from moving, or does the migration become less efficient over a certain distance? Is that possible to check?
Okay, funnily enough, these are some of the things that I have a project student working during the summer, looking into some of these questions. So one of the main questions that was like, I think it's to answer your question, my hypothesis, because I haven't tested this, but I've done something closer to similar to this, which is when you become a big aggregate, when you have an affinity for each other, then energetically it's favorable for it to basically form this cluster. But at the same time, being a larger cluster also hampers your mobility in some sense.
Okay.
So there is the force that is pulling you towards its destination, and then there is adhesive affinity for you to form this cluster. I do think there is going to be some sort of a tug of war between these two things that basically one probably slightly gives an advantage to a single cell migration, and in the case that is when you want a cluster migration. And as a consequence, I think there's probably a limiting size as to how big their cluster can be. Maybe there is an optimal range of the number of cells that can be in a cluster where you are moving pretty fast, but at the same time, you're not like where you take care of the energies by having boundary pixels in contact with cells of your own kind. At the same time, you can also move without much. I think this gets very, very interesting when you have an external signal that is very noisy, because in essence, when you have a cluster of cells, you can actually sense the gradients, direction of the gradient better, because you have a larger surface area, which in principle allows you to tell the direction of the gradient better.
Correct.
None of my simulations here had like chemoattractant that was noisy. But I think if you include noise, I think the process becomes even more interesting.
It would also be interesting because I was thinking particularly in terms of the size limitation for highly metastatic cancers. So are inherently those clusters that break off from the main tumor smaller in nature? Is that why they are able to metastasize more efficiently versus, let's say, so pancreatic cancers don't metastasize very well. I mean, it's known, but lung cancers do. So I was wondering if anybody has done any kind of simulation to see if the cluster size and the metastatic potential of that cluster is there any relationship between them? So this model sort of reminded me of that.
No. I mean, I can certainly. I mean, you can. I mean, I did phrase it in the context of cancer, but collective migration being such an important part there, especially because of metastasis. I completely agree with you, and more evidence shows that the conventional EMT where epithelial cells become this single cell is not true, but it's partial EMT where epithelial cells don't completely dissociate into single cells, but rather maintain these small clusters which migrate and so on. So I suppose there is a lot of advantage. I think one is that if you form a cluster, there is always a core of cells in the middle who are probably not affected by things that are in the environment that is trying to damage you. So there's probably a lot more advantages to being a cluster, but at the same time, if you have to go through some endothelium, which is very narrow space, being a big cluster is probably a bad problem for you because it's too narrow. So I think quite a lot of factors. I think it's a very interesting, many factors affecting whether you want to be part of a cluster, how big the cluster should be. So
yeah.
Thank you.
No problem. So any more questions? Okay, if not, let's thank the speaker once again.